thank to all organizing committee, and I would like to express my gratitude for all the help in this during these hard days. Thank you very much. We are grateful.
You're welcome.
Today, I would like to talk on brain oscillations as a probe of cognitive dynamics in degenerative conditions. I would like to talk on electroencephalography, electrical activity on the scalp that has been shown to represent macroscopic activity of surface layer of the brain underneath. And we know that electrophysiology basically can be divided into two, as resting EEG and task-related EEG. And from 2017, electrophysiologists have accomplished a big step because in 2017, 17 dementia with Lewy bodies had an electrophysiological biomarker. But this is not prevalent for Alzheimer research. But our aim is to put EEG into this frame for, I don't know when this will be happening, but we are trying very hard. Electrophysiology has lots of advantages because it has a great time resolution, much more than fMRI or other imaging techniques. It's non-invasive and not costly, and it is used less than it deserves. We know that EEG is a neurodegeneration marker because resting EEG correlates with MRI, structural MRI volumetry, and it can predict A beta levels. And this also is our previous work showing event-related oscillations also reflect neurodegeneration. And it is not specific to Alzheimer's disease. This is the basic flows of EEG at the moment, because PDD or DLB has similar findings. So it's not specific to Alzheimer's or PDD or DLB, but it is very sensitive for cognitive impairment. And as I told you, vascular dementia is also similar, has also similar findings. 2 years ago, we have shown that CSF-proven early onset Alzheimer's patients, when you use frontal theta over alpha, has a biomarker specificity and sensitivity. And you can just easily find and use it as a reliable biomarker for those early onset Alzheimer's. And these people can be hard to diagnose at the first place because they generally go to psychiatrists for mood disorders and depression, and it has a very hard differential diagnostic procedures. So event-related potentials and event-related oscillations help understanding brain dynamics, and they are sensory, motor, emotional, or cognitive task-related potentials, and they can give information about related networks. And among the teams, Greece has great neurophysiology teams, and I love this diagram showing longitudinal changes in mild cognitive impairment and a nice formulation of mathematical formulation of event-related potentials showing 200 decrement and latency increase over the years. And moreover, when you add CSF beta amyloid and P300, you can reach almost you can reach 100% sensitivity with about 90% specificity, which is great. Other than that, you can do a frequency analysis in event-related potentials, and you come up with event-related oscillations. And it shows that gradually decreased delta event-related oscillations in Alzheimer's and MCI patients. What's more, you can talk about the networks. Here we can clearly see sensory networks are normal in visual sensory networks are normal when you give a simple sensory task without any cognitive demand. It's normal, but when you give a cognitive task, which is a simple oddball task, you can clearly see the difference between the groups, and it indicates abnormal visual cognitive networks. And over time, you can see that in longitudinal studies, stable MCI has higher delta event-related oscillations than progressive MCI, reaching about 31% difference at the baseline. And when you follow up those progressive MCIs, they have a further 30% decrement in 1 year. So it tells us the progression and the detection helps detection in AD and related disorders. Furthermore, it also reflects neurodegeneration in PD, Parkinson's disease, MCI patients, because P300 amplitude is decreased, and it is quite strongly correlated with reduced putamen volume in those patients. And when you compare it with other methods, such as MRI, through deep learning, we showed that AD and healthy controls can be separated by 90% accuracy by using MMSE and global theta of resting EEG. And it is not prevalent for amnestic MCI and healthy controls separation because this discrimination was not very strongly made by either EEG or MRI, and it was below 70% by deep learning. So it wasn't very successful at differentiation of amnestic MCI, the resting EEG. About the connectivity studies, that's a very nice paper of Laura Bonanni showing hyperconnectivity in dementia is at the earliest stages occurs in anterior hubs. So it's for some reason there is a hyperconnectivity at the beginning of MCI stages, and then when the disease progresses, there is a posterior hub loss, and with advanced structural atrophy, connectivity occurs. So EEG informs us about the pathogenesis, overexcitability, and hypersynchrony, because we can see silent epileptic seizures, slow wave activity abnormality in REM sleep, and by seeing abnormal K complexes. And these occur about 10 years prior to clinical findings. In this overview, from now on, I will overview, the research teams in 2022 try to update what's going on in this field. And I would like to thank Dr. Josephine Cruzel, who did a year in review webinar. We were all panelists. And I have 4 items to go on. And the first one is neural overexcitability and dementia. And microglia has a role in brain rhythms. TREM2 mutation induces microglial loss of function. And when you have a TREM2 mutation in mice, and when you do a pontis oralis stimulation, you come up with hippocampal theta bursts responses. And those mice have lower theta oscillations in this regard. And in patients with AD-MCI, we see epileptiform EEG activity in some, and if you separate them, you can clearly see abnormal delta increase in patients with MCI and epileptiform activity. This is another beautiful paper, CSF, by Panhellenic Conference, showing quite quiet vigilance. Resting state, eyes closed EEG rhythms reflect quiet vigilance. And in patients with AD and epilepsy, and AD without epilepsy, you can clearly see slow waves. Synchrony increases across the groups. AD without epilepsy has less, something in between, but AD with epilepsy patients have higher, slower waves synchrony and lower alpha synchrony over the posterior region. This is another beautiful paper by Panhellenic Conference, and it shows us egg markers and brain excitatory and inhibitory balance and related neuropathology. They did MEG, and also they did tau PET and amyloid beta PET. And they showed that there is an increase in inhibition at the beginning of disease progression, and it is correlated with A beta, not tau. So at the beginning, we see increase in inhibition related to amyloid beta, and later in the stages, we see amyloid. I'm sorry, this is amyloid beta, increase in inhibition, and this is in the later stages of disease, increase in excitation related to tau. So this is the basic mechanism why we see hyperexcitability first, and then we end up with hypoconnectivity. The same thing holds true for sleep. Excitatory and inhibitory imbalance during sleep in AD ends up with epileptiform discharges and memory transformation disruption, and increase in cholecystokinin cell activation. Our group's latest work also shows hyperconnectivity occurs in theta event-related oscillations in MCI. If you kind of see what's going on, MCI has the highest connectivity, and then healthy controls have something in between. And at the very end, Alzheimer's disease dementia patients have less connectivity than healthy controls in theta event-related oscillations. So we think that physiological markers of cognitive systems in AD includes oddball event-related oscillations in AD, MCI, and ADD patients. Moreover, posterior resting state EEG alpha reactivity is decreased in AD, and also it decreases more in DLB, patients with Lewy body dementia. And we have to take care of or consider gender differences in many algorithms. For example, if you do a machine learning algorithm, you have to consider gender because it matters. As you can see here from the resting state activity in Alzheimer patients and normal elderly, you can see the females have higher alpha sources activity, and same holds true for event-related potentials, both in auditory and visual oddball paradigm. We have higher amplitudes in females. What's more, we are moving on EEG markers in clinical settings, and we see stable cortical excitability and repetitive transcranial magnetic stimulation shows TAPS as markers, and it has 24 TMS response, and it's stable in AD patients treated with repetitive transcranial magnetic stimulation over the precuneus. So it has a long-lasting effect if you do TMS to those patients. What's more, you can see the changes in behavioral FTD. You can use heart-evoked potentials in BV-FTD, and we know that they have problems in insula, amygdala, hippocampus, and anterior cingulate cortex. So they have more negative heart-evoked potentials, and that could be associated with cognitive profile, brain volume, and functional connectivity, shown by Birba et al. So we can use EEG markers in clinical settings using multimodal features for dementia classification, such as BV-FTD versus healthy controls, AD versus controls with great RAC analysis results, AUC results of over 0.90, and BV-FTD versus AD by using multimodal markers such as neuropsychological testing, demography, functional connectivity of EEG, structural MRI, and MRI functional connectivity. So if you put everything together, you have a very nice objective way of dementia classification. And right now, there are many studies trying to harmonize all these multimodal features. And it also gives hopes for treatment options for AD, because the potential increase in epileptiform activity should be monitored by EEG, and long-acting and sleep-stabilizing anti-seizure medication can help a lot. And there are many studies going on at the moment. They are trying to improve epileptiform activity and thus improve sleep disruption, and that would lead to better memory consolidation. For the treatment part, there is gamma modulation using sensory stimulation studies that lead. There is another paper, or two papers actually, structural and functional MRI and memory performance after that gamma entrainment showed better results at month 3. Also, our colleagues from Istanbul has also showed slowing of theta oscillations with using TOX, alternating current stimulation, improves memory. And you need to use individualized theta, and you have to find first who the best theta for that person, and you can use individualized theta alternating current stimulation, and that ends up with better memory improvement in patients, in healthy controls, I'm sorry. So induced gamma oscillations also promotes functional connectivity and improves cognitions in Alzheimer patients after TMS treatment. And you can also use event-related oscillations as a treatment effect monitoring, such as here, the patients with healthy controls have nice phase-locked event-related theta oscillations. And de novo AD group has kind of chaotic, and cholinesterase inhibitor treated AD group shows nicely oscillating theta oscillations at left frontal. And also, you can use nonlinear EEG studies, such as permutation entropy-based EEG studies, and it shows us the effect of memantine or cholinesterase inhibitors. This is the cholinesterase inhibitor study, and this is the dual therapy results, and there are clear changes between the groups. So I'm moving to international initiatives. So there are many continued efforts towards data sharing and harmonization, and there are many calls for consensus. And here you can see the international initiatives papers by Prado et al., by Parra Rodriguez et al., by Lipnitsky et al., Engemann et al., Lee et al. So you just go ahead and those papers, and then you can be part of these international initiatives. So to conclude, ISTART, AIC, Alzheimer Association's electrophysiology working group proposes physiology P markers in the ATN model of AD, and not ATN, but we propose ATPN EEG to be in the ERP. And would you like to become a member and check for on-demand webinars of ISTART? So I would like to thank you all, and thank you for this kind invitation. And I would like to say again, my thanks. And namaste, gonomones. Thank you.